single nuke RNA sequencing is a fairly new and powerful technique that provides us insight into gene expression at the single cell level, even from the complex tissues. And apart from single cell sequencing, it avoids this stress induced by dissociation of cells, which can cause artificial gene expression. So it allows us basically to identify different cell types, to understand cellular heterogeneity, to explore gene expression dynamics within individual cells, and it can reveal unique transcriptational signatures of various cell populations. This information can help us understand developmental processes, disease mechanisms, cellular responses to stimuli or treatments, and it can enable us to study even rare cell populations and spatial organization within tissue and cell-to-cell communication, which is basically what we did in our research. So we thought it would be interesting to combine single nuke RNA-seq data with our histological staining. And for that, we use this fairly simple technique called iterative indirect immunofluorescent imaging, where you basically stain your tissue for a lot, so to say, proteins. So we stained it for about 40 to 50 proteins. So we combined this in a way that with single nuke RNA-seq data, you get these high parameter cluster. You can do high parameter clustering. You have, in theory, the whole transcriptome, but you're missing spatial component to that. And with our histological staining, you can do low parameter clustering, as of course you do. You can do low parameter clustering because you stain for less proteins, but you get spatial component. So by combining these two, we could then localize single cell defined clusters within our lesion tissue. And by that, we could then define their spatial interactions. We could define their functional interactions. What we discovered there was quite interesting. We analyzed chronic active and post-demyelinating lesions. And we found that usually the most interactions that we found after conducting this computational pipeline of defining spatial clusters within tissue and then defining their receptor-ligand interactions, we found that most of these interactions across the lesion zones were mostly pro-myelinating. However, in the lesion center and the lesion rim, we found a lot of these myelin inhibitory and even cytotoxic interactions in lesions center. In post-demyelinating lesions, we found substantially less pro-myelinating interactions, which is kind of expected because inflammation is kind of ongoing in this type of lesion. So we hypothesized that maybe these myelin inhibitory mechanisms somehow override these pro-myelinating ones, and that is what drives remyelination failure in these cells. Then we did pathway analysis to see what are downstream pathways that are activated across these receptor ligands that we discovered. We looked into these pathways after that, and we kind of looked into their centrality in terms of what are the genes that are central in these networks that are involved in these pathways. And it, in the end, gave us two potential pathways that are serotonin receptor signaling and G-protein 37 signaling, which we are kind of which we are going to further confirm. Next steps are basically to investigate more lesion types. So we need to investigate remyelinating lesions. We need to investigate chronic silent and some control tissue as well. And apart from these pre-myelinating oligodendrocytes that we were focused on in this research, in this project, we will also look into this other subpopulation of control-enriched oligos as defined by one of these single nuke RNA-seq datasets by Yale. So these are the oligos that are also remyelinating, but are enriched in the controls. And we looked into ones that are enriched in the MS tissue. And also, we would like to look into these senescent cells, senescent astrocytes, as those were one of the as substantial part of cell interactions with these remyelinating oligos were with senescent astrocytes, among others.